In Larry Nieder's absence, my name is Kimberly Patterson. In Larry Nieder's absence, it's my pleasure to introduce our speaker today, Dr. Lena Hileman, who is the professor and chair of the Department of Ecology and Evolutionary Biology at the University of Kansas, as you can read right there. Her research integrates multiple experimental approaches to discover patterns and processes of evolution in flowering plants. What are you doing with your life? Wow, I got to get busy. Her research has been continuously supported by the National Science Foundation and has been shared in over 45 publications. In addition, Dr. Hileman is devoted to mentoring the next generation of biologists, and this commitment has earned her many awards and accolades. She will speak to us today on floral diversification in the Wildflower Genus Penstemon. Please join me in welcoming Dr. Lena Hileman.
Thank you.
Thank you.
Thank you. Very kind introduction. Let me get myself wired up. Okay. Is that? I feel like I'm not amplifying with it right here. I just sent something out to the group. Probably in the room. You'll be okay. You try to get it a little more centered. Okay, I think that they could hear before, they can probably hear now. I can hear the amplification. Okay. Well, thanks for having me. This is exciting to talk to a group of, mostly I'm assuming non-scientists, and I love to try to share my research. It's actually very difficult. Try to take it down to a level where hopefully this is accessible to all of you. It's a good challenge for me. And I want to say thank you all too, because I teach a number of classes at KU that are centered on floral diversity and flower development and genetics. And many of the gardens that you all, I think, volunteer, or at least even if you're not volunteering, you maintain, especially the butterfly garden over by Monarch Watch, are a rich and really important source of floral diversity for my class. So thank you very much. And if you see me poking around, picking flowers, it's for a good reason. So I'm sorry, I'm really just not gonna be able to stand behind the podium if that's okay. Yeah, it's not constructing what people see at home like it is on the screen. So do you want, we can take a second. I don't think I'm like going to be pushed for time. You want to try to? I'm trying. Are they seeing the full screen at home? No, and they're seeing. So they're not, you're not mirroring to Zoom the. Here's me trying to screen share, so I don't. Yeah, so screen share, try screen 2. And then see what they say. Yeah, okay. Okay. And then are they seeing? You have to share the right screen.
Are they seeing the right thing now?
Yeah.
Oh, okay.
But it's not advancing for me.
I'm gonna put it back on.
Oh, wait, yeah, go to presenters, slideshow. And then either any presenters of you is fine. I don't need. Oh, it's working here. Is it advancing? Change there. Okay.
Well, I guess that's all that matters. You just what you're seeing.
Yeah, I'm fine with, as long as they're seeing that.
Yeah. I don't know why.
And they're like, okay, super. Okay, we're on track. Okay, so I think probably you all are familiar with one of our most common penstemon sort of garden varieties, Penstemon digitalis. I don't study penstemon because it's very rare among penstemon in that it's not a diploid penstemon. So most penstemon, just like all of us in this room, are diploid. We inherited one chromosome from our mom and one from our dad. Most penstemon have that same pattern of inheritance, but Penstemon digitalis is a dodecaploid. So it has 12 copies of each chromosome. And that makes it very difficult to use as a genetic system. It may be why it's so, prolific in our gardens. Okay, but oops, now I am not advancing. Not advancing anymore. It's advancing here, but not there.
Usually you just have to click on the one and then it works. So you're gonna have to have the right one up.
Yeah, it's
Um,
sorry, we had a map.
today, so that's a little bit of the yeah. Going better than last month.
Okay, so far,
Okay,
we're
all right.
not gonna touch anything now. I'm gonna stand far away. Okay, but you also they may be familiar with other species that grow locally, well, relatively locally in the Midwest. Penstemon covea is the focus, well, I don't know, it's not advancing again. Let's see if it'll advance here. No. No, do I have to like fix the error? Yeah, it's working. I'd be happy to use them, but. This that popped up. Okay,
Go ahead. Let me try to advance it.
my video actually even loaded. Okay, let me try first, and then if it doesn't work, we'll say. Okay, so, we are doing some work in Penstemon covea. And, this is a picture I took of one of the individuals where we are doing our research on Penstemon covea. We're looking at how variation in the size of the reproductive organs, the stamens and the pistil, how variation in the size of those organs affects pollinator behavior. Well, actually, affects pollination activity, like whether those plants are achieving outcrossing to other individuals in these populations. And so I have a little video here of a carpenter bee visiting a Penstemon covea plant. Going after that. But I'm not going to tell you, this is still in progress, so I'm not going to focus on this. I'm going to focus on some other work that we're doing in my lab. And so first I want to just introduce you a little bit more broadly to the genus. It is the most species rich, has the largest number of species of any genus of flowering plants endemic to North America, with nearly 300 species. Most of that diversity is centered in the western part of North America, from Mexico through Canada. High diversity in the Rocky Mountains and Sierra Nevada and between in Utah. And we know from work from others, not from my lab, but from other labs, that the genus originated. So the ancestor of all living Penstemon today lived about 3.5 million years ago. And that around 2 to 2.5 million years ago, there was a really huge burst of diversification. So birth of new species of Penstemon that occurred around that time. And really for us for me as an evolutionary biologist, that's really recently. So there's a huge recent burst of diversity in this group, which actually helps to facilitate some of the research that we do. So I like to think of Penstemon as kind of divided into two categories. Most species of Penstemon are adapted to bees as pollinators, but a smaller subset, about 16 to 18% of species diversity in Penstemon, their flowers are quite different. You can see an example on the right in Penstemon barbatus. They're adapted to hummingbirds as pollinators. So I want to dig in a little bit into those differences in flowers between the two types of species we see in Penstemon. So Penstemon that are adapted to bees as pollinators have certain attraction and reward traits that we in my lab find really interesting. The main thing is the difference in color. That's really easy to see. The bee adapted species tend to be bluish purple. They produce a pigment called delphinidin. And the hummingbird adapted species tend to be red to orange, and they produce a very related pigment, a different pigment called cyanidin or pelargonium, other two reddish to fuchsia pigments. They also reward their pollinators with different aspects of their nectar. So the bee adapted species produce a very small volume of nectar so that bees will just get that little bit and go on to the next flower for pollination. And the hummingbird adapted species produce a larger nectar, a larger reward of nectar larger volume of nectar as a reward. So the hummingbird visitors who then require that larger energy source to like be attracted to come back to certain flowers. They're not going to revisit flowers that have very small nectar. They're pretty smart that way. Beyond just these attraction traits, there's also lots of traits associated with the fit of the pollinator to the fit of the flower to the pollinator to achieve really precise pollen transfer in an interspecific or an intraspecific way, in a way that's moving pollen from one flower of a species to another flower of the same species and getting that pollen to be positioned correctly to fertilize ovules and produce seed. And that those traits in Penstemon have to do with the overall shape of the flower. So bee-adapted species are shorter and fatter. Hummingbird-adapted species are longer and narrower. Here, the hummingbird, only its beak and tongue can kind of get into the flower to get that nectar reward. And the pollen is going to be the top. Oops, find the pointer here. The pollen is going to be deposited right on the hummingbird's forehead. We can see the reproductive organs sort of sticking out of the top of that flower. And so those reproductive organs are also different, quite different in their length, with much longer reproductive organs, stamen and pistil, male and female reproductive organs in the hummingbird-adapted flower, and much shorter organs in the bee-adapted. So it's like a little overview of what we call the pollination syndromes. So it's a suite of traits that are kind of complexly integrated with each other that allow for the attraction and efficient movement of pollen by a particular pollen, what we call pollen vector, which is the bee or the hummingbird. So we have used DNA sequence data, so genetic data, to look at the relationship among a large number of Penstemon species. And basically what we're able to generate with that DNA sequence data is what you can think of as a family tree. And this is over 100 species of Penstemon on this family tree. So let me explain what we're seeing here. The detail, I just want you to kind of give it a general overview. We're seeing species with dots at the tip that are each other's closest relative. So for instance, these two species of Penstemon are each other's closest relative. One is bee-adapted with purple and one is hummingbird-adapted in red. And they share a very recent common ancestor that's inferred. This is an inference. We don't see that ancestor, but we infer that that ancestor was adapted to bees as pollinators. And so one of the first really important takeaways from this work is that all of the hummingbird-adapted species are not each other's closest relatives. They did not evolve one time. So it's not that there was one transition to hummingbird pollination and then a whole bunch of new species arose from that hummingbird-adapted ancestor. Instead, what we see is that scattered all the way around the tree are these independent transitions to hummingbird pollination. So this seems to have happened again and again and again. And on this tree, which is only about a third of all species diversity in Penstemon, we find 16 transitions to hummingbird pollination. 16 independent times, Penstemon lineages have switched from bee pollination to hummingbird pollination. If we sample the rest of the species diversity, we find even more changes, I think, to be the pattern in this group. So this isn't unique to just Penstemon. In the western flora of North America, we see many transitions from bee to hummingbird pollination, and this seems to be associated with a large diversity of migratory hummingbirds present that migrate across western North America. We see this in the plains too, but the hummingbird diversity species here is much lower than in the western US. And so these are just some examples where in other lineages, other genera, not Penstemon, there have been these transitions to hummingbird pollination, and many of the same traits are involved, floral traits. Okay, so when we see these repeated origins of a particular trait in evolution over evolutionary time, we call this convergent evolution. Convergently, many lineages of Penstemon have converged independently on this ability to attract and use hummingbirds as their pollinators. And so that's what we see it kind of scattered in multiple places across the tree, like I showed you on that circle tree or family tree for Penstemon. And we get pretty excited about this because these independent origins tell us that there's something about the environment, in this case, the presence of hummingbirds and their efficiency in moving pollen, that is a selective pressure for adaptation to push lineages in that direction again and again and again. Like if there's hummingbirds, to speak anthropologically, if there's hummingbirds, the Penstemon want to use them. You know, they want to get to that space of floral shape and color where those hummingbirds will visit their flowers and move their pollen efficiently and increase their fit. So we now can use this family tree of Penstemon to answer a couple of really fundamental evolutionary questions. I'm going to share these with you in a kind of hopefully brief way. We want to know if it's just as probable, just as likely for transitions from bee adaptation to hummingbird adaptation to occur as it is for transitions from hummingbird adaptation back to bee adaptation. Is it like, are these things just moving back, are these traits moving back and forth at equal rates over evolutionary time? So there's just this dynamic of like transitions back and forth. And we could use the family tree of Penstemon we built and do some modeling, which I won't show you the details of, to answer that question. And the answer we see is that indeed those transition rates are not equal. So this is probably not surprising from everything I've already said. But now mathematically we can prove that it's really likely for these transitions from bee adaptation in the big fat green arrow, that's that rate of transition to hummingbird pollination, that's really high, and there's a really high likelihood of movement in that direction, but an actually nearly zero, like all of this density on this plot is pushed towards zero, almost zero movement or evolutionary transitions back to bee pollination. So it's kind of weird. Why would there always be this direction towards hummingbird and no transitions back to bee? Well, we think, and we want to ask about this, I won't get into it now, but we think that there's reasons about the complexity of how these traits are assembled into pollination syndromes within the flower that actually make it very difficult to step. It made it easy to step from traits that are associated with bee pollination to hummingbird pollination, but very difficult to assemble those traits again into bee pollination. So that's a very much harder set of steps to take evolutionarily. Now let's think about this for a minute, a second. If there are many transitions to hummingbird pollination, like you can always go in this direction to hummingbird pollination, but you can't go back to bee pollination, and we play the evolutionary tape of time, what should Penstemon look like? Penstemon should be mostly hummingbird pollinated, right? If you're always going in this direction, you're never going back, and that's that bias towards hummingbird pollination, but that's not what we see. Like 15, 16, 17% of Penstemon are hummingbird pollinated, but most of them aren't. So it's possible that maybe the evolutionary tape of time just hasn't played long enough. I told you that the genus Penstemon is a really young genus. It's only 3 million years old. So maybe if we waited 10 million years, all Penstemon would be hummingbird pollinated. Then an alternative to that is that maybe there's some other process happening that actually counters this rate of transition to hummingbird pollination. And that other process could be the rate at which lineages in Penstemon diversify. So what I mean by diversification is a lineage or a species, its ability to produce new species, to split and make new species, to split again and make new species, and to not go extinct. So diversification in evolutionary sort of lingo is the rate at which you can speciate, make new daughter species, kind of split and split and split again, and not go extinct. That would be a high diversification rate. A low diversification rate might be not being able to split into new species or rarely split into new species. Or you split, but then you're just going extinct. Those species are going extinct so quickly that there's this low rate of overall species diversity. So we could test this, and these are cartoons meant to illustrate these splitting events, so speciation events here in a bee-adapted lineage, and some extinction, or these speciation events, these splits in a hummingbird-adapted lineage, and then some extinction events. And in this little cartoon, they're equal to each other. So we tested whether they are actually equal to each other, and we found that they're not equal by a long shot. And that actually those lineages that are finding themselves in that bee-adapted state, they keep producing new species. And those species don't seem to go extinct very quickly. But those lineages that find themselves in that hummingbird-adapted state, they're either not speciating very readily, or they're going extinct really at a high rate. We don't really know which one of those it is, but we have some ideas of how to test those different alternatives. So what I conclude from this is that it's not, and actually our models tell us, it's not that we need to wait 10 million years. Actually, Penstemon have achieved an equilibrium where if we look at the rates of transition to hummingbird pollination and the rates of diversification within those two types of lineages, bee and hummingbird adapted, even in 10 million years, the balance of those two processes would lead to diversity in Penstemon where about 15 to 20% of species are hummingbird pollinated and the rest are all bee pollinated. So we're at that equilibrium right now. So that does suggest, or that equilibrium state is dependent on no changes in bee diversity, habitat diversity, hummingbird diversity. Of course, it's totally dependent on the ecological setting, but right now, our evidence suggests an equilibrium that could be disrupted over the next 10 million years, or even maybe the next few hundred years. Okay, so I'm going to change gears and talk a little bit more about some more focused genetic work that we're doing in my lab. And in the more focused genetic work, we are trying to understand, how do these transitions happen? How do we get a flower that's adapted to bees as pollinators? How does evolution proceed to lead to this shift to hummingbird adaptation syndrome, the shape and the color and the nectar report of those flowers? And so I'm going to share with you a little bit about what we're learning about the genetics of those transitions. Okay, so I'm going to introduce you to 3 ideas that we have about these are sort of our hypotheses, why might it be easy to go from bee-adapted to hummingbird-adapted pollination syndrome. So over here on your left, from that bee-adapted state to these independent convergent origins of hummingbird adaptation. What about the genetics might make that easy? So first, it might be that there's very few genes that are required. So let's say, for shifting from blue to red flowers, if 10 genes have to change how they function, that's going to be a lot harder to achieve evolutionarily. But if only 1 gene has to change, then that's going to be much easier. Maybe that gene can change repeatedly in this case, this example of these 4. Maybe that gene can easily be changed one time, like in each of those lineages. So that's what we consider low genetic complexity. Just a few genes or 1 gene is required to change a trait and not many. Alternatively, maybe the genetic variation is readily available. So one of the things we're interested in, and I'm not going to talk about today, is hybridization. Most Penstemon species, especially if they're adapted to bee versus hummingbird, if they're like each other's closest relative, and one's bee adapted and one's hummingbird adapted, they make hybrids in nature. They are not reproductively isolated by any other mechanism, so they can, and we take advantage of this in the study initially, this ability to make hybrids. Some species pairs do this a lot in nature and have what we call hybrid swarms or hybrid populations, and others you'll just every once in a while, rarely, but sometimes find a hybrid in nature. So that would mean that with hybridization, hummingbird adapted genes could move into bee adapted populations. And that might facilitate these transitions. And then maybe these traits are really highly correlated with one another. So let's say if you shift tube width, that's automatically going to be correlated with corolla tube, the flower tube. If you go from fat to narrow, that forces the flower the tube to just get longer somehow. Somehow those are correlated with each other, or the genetics of that are linked, and they're not independent changes. So that would also make it easier to shift if you don't have to have, if these traits are correlated and you don't have to have multiple genetic changes underlying each individual width and length bit of that variation. So these are our three hypotheses for reasons why these transitions might be pretty easy. Okay, so to test those hypotheses, we really dig into one small group of penstemon that we have developed as a kind of powerful model system to use genetics to address these kinds of questions about how flowers shift their shape and rewards and color. And that system is made up of basically two species. Penstemon barbatus, which is hummingbird-adapted, and then we work mainly with Penstemon neomexicanus, but it's part of this larger species group called Penstemon virgatus. They're not really distinct species, they're kind of nested within each other. So I'll talk about Penstemon neomexicanus as our bee-adapted species in this model, and Penstemon barbatus as our hummingbird-adapted. And these two species are found co-occurring from across the Rocky Mountains into New Mexico and across parts of Arizona. You can see with the blue dots, there are places where Penstemon, this is virgatus, but Penstemon neomexicanus and virgatus are not co-occurring with the hummingbird-adapted barbatus. Places in purple where they're co-occurring, and then in red where the hummingbird-adapted species barbatus is found by itself. So those dots are all just individual populations we've looked at, or mapped from herbarium collection material and just put onto a map. Okay, so back to our family tree. These two species are each other's closest relative. Here's our barbatus tip on the family tree and our neomexicanus tip on the family tree. And they share a recent common ancestor that was likely similar in its flower shape and color and reward to Penstemon neomexicanus. Okay, so here's where in my lab, my students and I, we put on our Mendel hat and we do plant genetics. So some of you may remember this from college or high school biology classes. Mendel worked with peas, and he was looking at the inheritance of, the purple or white variety of flower, or the green or yellow peas that are produced, or the wrinkly or smooth peas, and what the inheritance pattern was of those traits. We're doing exactly the same thing, but we're looking at the traits in the bee-adapted neomexicanus and hummingbird-adapted barbatus, but we're crossing them and producing progeny just like Mendel. We have the advantage of being able to sequence genomes. Mendel couldn't do that, and this takes our research to a bit of a different level than Mendel could go to, but the fundamentals are the same. So fundamentally, and just like Mendel, we make a hybrid between the neomexicanus and barbatus parents. We call this the F1 hybrid, or filial 1 hybrid. You can see that the flowers are somewhat intermediate in their overall appearance between the two. We allow this F1 hybrid plant to self-pollinate. We don't just allow it, we go in and we move pollen and we force it to self-pollinate. And we collect a bunch of seed, which are the grandchildren of these parents. And we call those the filial 2 or F2 population. And what happens in the F1 individual during the production of the pollen and the ovules that will produce that seed when we self-pollinate is a whole bunch of genetic recombination among chromosomes happen. So it mixes up all the genetic material that was found in this parent and that parent, so that these offspring are a mashup of like color and width and length and reproductive organ length and nectar volume. It's all mixed up. This is just a little like a set of flowers that could hopefully show you a little bit of that mix up, that variation that we see in the F2. Okay, now we can put some chromosome information on this, some genetic information. So now let's imagine we do this, so we don't have to imagine. We see the genomes of all of these individuals. So we have all the genetic information of these individuals, and we can track for any single one F2 individual its along its chromosome, whether it's inheriting pieces of its chromosome from the bee-adapted parent in purple, or pieces of its chromosome from the hummingbird-adapted parent in red. And these are diploids, so they'll have like different inheritance on each of those two chromosomes. And have any of you done like use 23andMe and looked at your, yeah, so on 23andMe, you can actually look at this kind of mix-up of inheritance patterns on each of your chromosomes and kind of trace where those might have come from. So in this example, we just have 4 F2 flowers. These 2 are really chunky and fat, and these 2 are really narrow. And when we look at the DNA, we scan it using software, not by eye, and we find regions where, in this case, these 2 fat flowers always have both of their chromosome pieces, or both of their chromosome sister chromosomes in this region are purple. And it's purple in these regions too. So that tells us that in this region of the genome, these 2 individuals inherited chromosome pieces from this parent. And these 2 are narrow, and you can see in the same region of the chromosome, they're carrying 2 copies that were inherited from the hummingbird-adapted parent. And so we're looking for regions where inheritance from hummingbird parents is associated with the trait narrow tubes, like the hummingbird parent, and regions of the chromosome where F2s are always inheriting chromosomal complements from the bee-adapted parent, and that's always associated with being Y. So that allows us to pinpoint exactly where in the genome is the variation between the bee-adapted parent and the hummingbird-adapted parent driving that difference in tube width. So in this case, it's this little black line here, so we could go back and say, well, right here on the chromosomes and here, there's some genetic variation, maybe changes in DNA between the A's, G's, T's, and C's found right here, and the A's, G's, T's, and C's found right here, that developmentally tells that flower to develop as a narrow flower versus a wide flower. You can really pinpoint those regions of the genome. So there's your genetics. It's hard to talk about the research without giving some of this background. So we do this across many hundreds of FGs. Usually in our population, we have 400 or 500.
Does it make any difference? Because obviously the flower could be a male, or the pollen would be from the male and fertilize the female. Does it make any difference which?
It doesn't, because these are the Penstemon flowers, just like most flowering plants, they're hermaphrodites.
Yeah.
So we can cross from treating the bee as the mother and the hummingbird adapted species as father, and the F1 looks exactly the same. And yeah, it's like whether it's like red and blue or blue and red in the other direction, it doesn't change the inheritance pattern at all. And usually it doesn't actually. Yeah, in flowering plants. Have we taken the questions for some clarification? Yeah, before we move on.
That little black line that you drew there, at the very beginning.
Right here.
Does it have to be there? I mean, if you move it up a little bit.
So anywhere else, I purposely drew this in a way that it couldn't be drawn anywhere else.
Okay.
So a line right here is like, yeah, it's red and purple. And then if we trace that down, it would be red and purple, purple, purple. So it would be, this individual is in this region, has both of its alleles from the wide parent, but it's narrow. So it would be like the opposite, would associate in the opposite direction. We're looking for associations where that inheritance from the particular parent with the particular trait associates with the trait.
Okay.
Yeah, we're really particularly looking for that direction of association. And there's a lot of statistical analysis behind it, but looking for that association. So we do this in the greenhouse. Here's just a little snapshot of some of these F2 hybrids growing near each other in the greenhouse. Wish we could have this talk in there. There's like lots of fun flowers. Yeah. And you're always welcome. Send me an email. Take anybody through the greenhouse who wants to. We have a public part of the greenhouse in here we're called to visit. Okay, so we can then pinpoint these regions. This was our hypothetical example, And this is now a cartoon of our real data. I'm not going to show you the actual real data. I'm going to show you what hopefully it makes it a little bit easier to understand. So we have found individual regions of the genome that where that variation that is inherited from the bee or hummingbird adapted parent really, really strongly predicts the trait of interest. Like there's a very strong association. And for 3 of the traits, there's only one place in the genome that has that strong association. Everywhere else in the genome is completely not associated with the trait. So just one place. And this is actually quite surprising. When you do this work in most flowering plants or any organism at all, there's usually many places in the genome that contribute to the variation you see in traits, not just one. But in this case, we see on chromosome 1, there are 8 chromosomes in Penstemon, and unless they're there are 8 chromosome pairs, and we see on chromosome 1 a region on what we call this left arm of chromosome 1 in the red arrow here, a region that the variation here almost perfectly predicts color variation between the 2 species. And a region right near it on this left arm of chromosome 1 that almost perfectly predicts the nectar variation between the 2 species, large amounts or small amounts of nectar. And we see a region over here on chromosome 4 with the green pointed to with the green arrow that very strongly predicts the differences between narrow and wider flower tube width between the 2 species. So this suggests that there is a single gene. We don't know, well, I'll tell you where we do know the genes, but in corolla tube width and nectar volume, we don't know the gene that's sitting in that position, but in flower color, we do know the gene. I'll tell you about that. But it suggests that there's one gene here, and one gene here that changes in that gene. Simple DNA sequence changes, G's, A's, T's, and C's, shape whether the flower is making a small amount or a large amount of nectar, a narrow tube or a wider tube, a red flower or a blue flower. And let's dig in to that example where we do know the gene for the flower color difference. So I told you at the very beginning, I gave you these complicated pigment names. Pelargonidin and delphinidin are the pigments that we see as red and blue in the hummingbird and bee-adapted flowers. And there's a complicated enzymatic set of reactions that result in these pigments. There's this gene right here called F3'5'H that is broken. It just doesn't function. It has loss of function mutations in the red species Penstemon barbatus. And when it's broken, when it's working fine, this pathway really moves a lot of the product down to make delphinidin. But when you break it, the only thing that can happen is that the pathway can go down this arm and make the pelargonidin. So that's a really simple change. It's really easy to break a gene. Like lots of mutations can break genes. So it's very easy for that. We think this is a very simple genetic change that's led from a shift from blue to red by breaking a single gene. And actually, we know that that gene has been broken at least 13 times in 13 different hummingbird-adapted species. So that has repeatedly, independently broken to make the red flowers. So when we get back to our hypotheses, that one example, this is just flower color, but it tells us that indeed that low complexity can help to contribute to transition to hummingbird population. So the other traits, tube length, stamen length, and style length are more complicated. So our results point to 3 different places in the genome where variation in presumably single genes at those locations affects the length of the corolla tube, and then the length of the stamens, the reproductive organs, and stamen twin styles. But when we lay these all on top of each other, we see that they actually stack up in exactly the same places in some cases. So this tells us that there's probably a single gene right here that if you change it, or a single gene right here that if you change it, it results in going from shorter tubes to longer tubes, but also longer stamens and longer petals, longer female reproductive organs. And so we can see this looking just at the correlation of these traits in the F2 population, where just as one example, if we go from short tubes to long tubes, each of these dots is a little F2 individual in our population. As we increase tube length, we also increase stamens. So they just are forced to move together. Probably because one gene controls both the length trait in the corolla and in the same. So this really points to this third hypothesis about trait formation, that that's part of we have support that part of the reason for the ease of transition is that these traits are for each other, at least in some cases. Okay, that was a deep dive into some genetics. But we're working with a number of these penstemon species doing this kind of work, looking to see if we see the same changes in the same places in the genome and the multiple origins of penstemon transitions to hummingbird pollination. And I think this is revealing some of the patterns and the processes that allow for repeated origins of hummingbird pollination. I will not drag you through penstemon genetics any longer. And I'll just go ahead and thank some of my collaborators, people who have made this work possible, especially my team members, undergraduate students, graduate students, and postdoctoral researchers. And with that, I'm happy to answer any questions you have to the best of my ability. I think watch the clock. Did I go? What time is it? Oh, there's the clock over there. Oh, great. Perfect. I hope to keep it under 45 minutes. I can get that. Yeah.
But the gene is random, it's not in response to the environment.
That's absolutely right. So mutations occur at random. And it's when we think about that F3.5 kinase gene, where it's a loss of function, where that gene is being broken to lead to red flowers, it's not that those plants want to have red flowers, so they break the F3.5 kinase gene. It's that mutations are constantly occurring every time we make egg or sperm. Every time the flowering plant makes pollen or ovules, those pollen and ovules have hundreds of new mutations just in a single pollen grain, in a single ovule that's produced and then potentially part of the next generation of that lineage. And so if one of those mutations happens to fall in F3.5 kinase and break the gene, and the offspring is red, and red attracts hummingbirds, and hummingbirds are this amazing pollinator, selection will favor that mutation, and it will increase in frequency in the population. If that mutation, on the other hand, prevents the plant from being able to make viable pollen, that mutation will not be passed on, selection will remove it from the population, and we won't ever see it again. So the mutational process is random, and then the selection process, natural selection, acts on that new variation, either to favor it or disfavor it.
Have there been any insights between, there have been sort of parallel studies on hummingbird population, pollinating?
No, I mean, yeah, so we don't see the rapid change. We do see a diversity of hummingbird species that seem to have some differences in like their beak length or their tongue length and things like that that may affect how they interact with pollinators. But the migratory hummingbirds that we see prevalent in Western North America don't show a lot of that variation. So they seem to all be able to visit any of, not just penstemon, but the diversity of hummingbird-adapted flowering plant species that we see in Western North America. In South America, it's a very different situation where there's a lot more diversity in the shape of the beaks and the length of the beaks and who the hummingbirds are visiting. And that's partly because in South America, the hummingbirds are mostly non-migratory. So they're sort of focusing and locally adapting to the flora that's available. And maybe partitioning that floral resource. Yeah.
Logical, mutation and expressive genes and such. Do different flowers have better mechanisms to identify? I.e., are there some flowers that evolve faster?
No, I think when we look at least within penstemon, the mutation rate and the ability to maybe like epigenetically like mask mutations is we haven't looked deeply into that, but we don't see any evidence that it's different among penstemons. I don't know if penstemon maybe is different than other flowering plant lineages. That's an area of research we haven't looked into.
It seems like, you know, when you look in your garden, there's a lot more bees that you see than you see hummingbirds.
Yep.
So the hummingbird is a super pollinator.
It's a super pollinator, we can talk about that too.
But is that one of the reasons why more of the red hummingbird flowers go extinct because there's not as many hummingbirds to?
Yeah, I think, exactly. I think what happens, so first of all, in Western North America, there are many more hummingbirds. You would run across hummingbirds and pollinators much more frequently than you would here. Unless you were like feeding your garden with a feeder, like you're training hummingbirds to come to your garden with a feeder. A large diversity of red tubular flowers, they kind of are cued in on. So I think what's happening, one of the things that's happening is that as long as the hummingbird pollinator population is high and they're efficient when they're there, as long as they're there, then these lineages will do well, they will stay in that hummingbird adapted space. But then if the pollinator, like if pollinators aren't migrating for a few years through that region, then their fitness drops really quickly. There might be plenty of bees, but it's this is what I was saying about the constraint and it's easy to go in the direction towards hummingbird pollination, but hard to go back to bee. And we think that's because basically if you're a bee adapted flower, hummingbirds are super promiscuous. You make a lot of nectar, they will come. That's all you need to do. So you can shift that nectar volume. That can be like maybe the first step. And they will come, and then maybe after you find 5 to 7 gets broken, and they're red, and they learn to cue in on that red signal, and then all the other shape changes kind of follow, maybe slowly. So that's easy to do. But if you're a very narrow tubed flower, you're sticking outside the flower, and you're making this large nectar reward, and there's no hummingbirds around, a bee might really want that nectar reward, but many bees won't be able to access it. Not in a way that efficiently pollinates the flower. They might use the nectar robe to the side of the corolla, or, you know, very small bees might get in and get the nectar reward, but then because the anthers are sticking out of the flower, they're not going to carry pollen. So there isn't a step to take. There is no step where, like, the bee goes to the hummingbird adapted flower, and there's one step that allows it to not take the next steps. It just, that first step isn't available. And so we have ideas about how to put those F2s out in natural settings, like in Arizona, where those species co-occur, and ask that if you're an F2 plant that has all the traits that are associated with hummingbird adaptation, but maybe one is associated, you have the Y, you have one piece that's like the transition back in the other direction, do you have high fitness if we exclude hummingbirds? We can test that, we just haven't done it yet. That's a long answer to an insightful question. Yeah.
This is kind of the flip side of that. So I know that, you know, we're very limited with the number of species of hummingbird here in Kansas, you know, for moving further.
Right.
But if our zones keep changing and we keep getting warmer, and let's say, you know, we don't freeze in seasons we used to as time goes on, and we switch from zones into, say, zones that we see down in Southwest right now.
Yeah.
Could that change not only the flora and fauna that we see growing, but could we start seeing new migrations of different species of hummingbirds start to move up into Kansas because we are now staying warmer, and our winters are not so much deep freezes as they are, you know.
And the freeze doesn't matter so much because they're migratory. These hummingbirds do move back, they migrate back to Mexico mostly in the winter, even if they're going to California. They're often very high elevation, maybe desert or high elevation migrators, but they're often still moving back south. But it's something, it's so interesting because I am so focused on like how our climate change is affecting plant distribution. As a plant biologist, I had never considered that could change the patterns and diversity of hummingbirds we might see here. And I think it could. I think that's an interesting insight. And that could then, you know, we have a few hummingbird-adapted Penstemon and other hummingbird-adapted species that are native, not very many, but we might see them thrive in this bit.
And well, it's interesting because my sister lives in Portland, Oregon, and I'm jealous because she has her feeders going and I have my feeders going.
Oh, it's a night and day.
And my feeders going is
Yeah.
this much time of the season. And hers is year round, but lately she's had more extreme heat and more snow than she ever had before. And so, you know, she'll brush off the snow and she has to have a warmer for her, you know, little, you know, cover for her hummingbird feeder to keep the snow off of it and so forth. So she's doing her best to adapt them. But what if they don't like that so much? How now they start shifting over?
I think that's possible. Yeah, definitely possible. And I don't know how that would, I'd have to really sit down and think about how that could affect, you know, the flora,
Right.
but it definitely could. Yeah. So do you all know why hummingbirds are such good pollinators compared to bees? Most people usually want to hear about that, you know, but do you have a sense of that? I mean, you all work in gardens all the time, maybe you have like a sense of it already, but when bees visit a flower, obviously they're interested in that nectar. But bees also really strongly rely on pollen as a protein source for their broods, whether they're, you know, honeybees, you know, colony or solitary bees, they're bringing that pollen, they're collecting it, putting it into their little leg bag, there's a special word for that, entomologists know, and taking it back to their brood to feed. So that's what we call pollen dismute. When a bee visits the flower, it's getting that nectar forward, it's moving pollen, but a huge amount of that pollen it's taking for itself. When a hummingbird visits a flower, it gets its nectar reward, the pollen is deposited on the head or forehead or beak, and it's just all moved for pollination. So they're much better pollinators. And we actually see in Penstemon some traits that we haven't genetically dissected, because they're just really hard. And maybe these will be some of the next things we look at. But if you look at that bee-adapted Penstemon or any of the bee-adapted species and the Barbados, the hummingbird-adapted species, the anthers, so we spend a lot of time looking at these flowers very closely, the anther sacs, when they hiss and release their pollen, in the hummingbird-adapted species, they just dump it all. So take the pollen, go, procreate. But the bee-adapted species, they slowly open their anther sacs a little bit. The pollen's much stickier. It sort of releases a little bit at a time. They're restricting. There's a lot of selection to like hold on to that pollen. Make sure if you dump it all, if you're going to just dump it all at once, the bee's going to get so much of that. Like, don't let that happen. Just release a little bit at a time. So there are traits like that we have some projects ongoing. In fact, I just got a large grant from the National Science Foundation to try and figure out more of these more subtle traits and their genetic basis that define these what we call pollination syndromes, the adaptation to one another.
Yeah, and you say that Penstemon specifically had it was unusual for it to be able to use the gene. And that's unusual.
Well, it's unusual. Okay, so it's really required for us to do this. It was necessary to be able to cross bee-adapted to a hummingbird-adapted species. We could not do any of, and we do this in multiple species pairs. I just showed you the Barbados mexicanus is our most well-developed model system. But we have to be able to cross bee to hummingbird-adapted parents to make that F1, and then the F1 has to be self-fertile to make F2. And that's not very common in any, you know, in any organism. Like you mostly can't reproduce with even your closest species relatives. But in Penstemon, largely with the bee and hummingbird-adapted species, because they're reproductively isolated, what we call prezygotically by the pollinators themselves. That means there aren't a lot of barriers after we can move pollen between them. So many of the systems that might be like maybe in columbines, you might want to do this. Look at the bee-adapted and hummingbird-adapted or hawk moth-adapted columbine. And some of those species are interfertile, but not as highly interfertile as Penstemon. Partly that's because we've had this recent rapid radiation. And some of those other lineages are a little bit older, and those species that are recently, or those species that have diverged and become bee and hummingbird, they actually maybe have diverged in more deep time. They have more time to accumulate what we would call postzygotic barriers, things that prevent the pollen from germinating, or for the F1 seed to be able to be viable. So we're kind of lucky in that recent rapid radiation, and then they have this pre-biotic isolation. Bee-adapted species are less likely to be, like two closely related bee-adapted species in Penstemon are less likely to, we're less likely to be overcrossed. Yeah, so there's a little bit of lopping off in all of this, just because the system has the history that it has. Well, thank you all. It was nice to share with you.